excellent panels so far, and ahead of us, I mean, and that saves us, having to tangle ourselves up with the convergence of targeted oncology with various FDA and congressional, pricing reforms. Thrilled to be joined on this panel by three innovative drug developers and seasoned drug developers as well: Jim Porter from Nuvalent, Steve Brady from Tempest Therapeutics, and Zach Hornby from Boundless Bio. And what we thought we would spend a bit of time on is, kind of the intersection of a few trends, chemistry advances, which each of these three leaders and their companies are certainly, sort of helping to lead this sort of chemistry revolution, I would argue, but really intersect that a bit with, biology insights. How do we think about, further improving on validated targets? How do we think about novel targets, de-risking, and the like? And then ultimately, kind of connect the dots from there in terms of how we think about the path forward, in terms of clinical development, even, possibly looking far enough into the future towards, sort of regulatory issues that are specific to this issue of targeted oncology. So those are sort of my opening comments and framework, but Jason, you want to maybe add a thought or two and kick us off with a question?
Yeah, sure. I mean, as we think about the precision oncology space, I think what historically has attracted investors is fast time to de-risking, right? In terms of being able to run a phase 1/2, get an overall response rate, and validate, some of the opportunity set. As the field has evolved, maybe this is a good softball question to start off with. Thinking about established targets, to the extent that there still are opportunities there versus the need to push forward into more novel targets. Maybe I'll throw that out there. Is the field of established targets pretty picked over? Are there opportunities to improve upon established targets through improvements in more selective molecules, drugs that have better coverage against resistance mutations?
Sure. Yeah, happy to take that. So Jim Porter from Nuvalent. At Nuvalent, we're focusing on precision oncology. The foundation of the company is a deep expertise in chemistry. We focus on clinically proven kinase targets. Clinically proven, the key there to Jason's question, our two parallel lead programs are targeting ROS1 and ALK, both which have multiple drugs approved for those cancers. So you would say, are those targets picked over? Is there clearly an opportunity? When we started the company, we went and formed relationships with physicians in this space that have worked on these specific earlier generation inhibitors targeting ROS1 and ALK, and understood from their perspective, what are the limitations of the current therapies, what are the needs of the patients that they're treating, and crafted very specific target product profiles that suggested there are needs that need to be addressed with next generation molecules that we could design through innovative chemistry. The tough part was really the chemistry. Could we get the chemistry to work to go after those targets? And now that we're in the clinical development with each of those two parallel lead programs, we're clearly seeing the interest from the investigators that started the collaboration with us years ago in defining those target product profiles, as well as the patient advocacy groups that have patients that have these specific diseases, recognizing that Nuvalent is trying to go after targets that are not being specific needs that are not being addressed by the available agents. So I certainly appreciate the comment and the perspective that many of those targets may seem crowded or picked over. Is there clearly an opportunity? I think at Nuvalent we very much believe there are. If there's patients in need, that's something that's of interest to us.
Yeah, but before we let you off the hook on using that phrase target product profile, because this is a topic I love to kind of expand on. I mean, there used to be a kind of a classical definition of that, which had to do with drug-like properties and kind of add-me features and the like. But what you're speaking to is kind of clinical unmet need, evidence that we have, translational evidence that we have coming from patients, coming from models that would say, okay, the limit on the existing therapies is this. So say a little bit more about how you think about target product profile when it comes to that, kind of the intersection of biology and then back to chemistry again. Like it's different than how people used to think about kind of what used to fit on one slide of target product profile.
It's a great point, Keith, and thanks for the clarification. So, yeah, what we're specifically getting at here is, what we sort of worked on with the physician scientists in this space is, let's give ROS1 as an example. The emergence of ROS1 resistance mutations. So there's currently no approved therapies that are active against a major ROS1 resistance mutation that emerges in about 40% of the patients that have been treated with first-generation ROS1 inhibitors. So that is a specific need that we're looking to try to address. In addition, the ability of the drug to cross the blood-brain barrier. So patients with lung cancer often, unfortunately, have disease that will spread to the brain. So having drugs that can cross that blood-brain barrier, that can treat patients with CNS metastases, is an important component of what we're trying to solve for through innovative chemistry. And the third area for our profile is that we're trying to avoid a specific off-target that is known that if you inhibit the specific off-target in the CNS, it could lead to a broad spectrum of nervous system disorders, things that physicians have told us are dose-limiting for their patients. And if we could specifically design against that, use innovative chemistry to design against that, perhaps we can keep patients on therapy, hopefully drive deeper and more durable responses with a next-generation inhibitor.
Great.
Just to your point on the TPPs, I agree with you. Ours are the same. Steve Brady with Tempest Therapeutics. And all of our programs, to your point on picked over versus going completely novel. So we have 4 programs ranging from completely novel target to our knowledge, no one is working on, all focused in oncology, all the way up to phase 2 randomized data. But our 2 clinical programs we had very different experiences with because we're not a platform-based company, so our target selection doesn't come out of a specific biologic hypothesis or a pathway or anything. We literally go look for targets that we think are really interesting and could make a difference in patients' lives. And so we do start out with the where's unmet medical need and how can we prosecute and do these 2 things meet? Our lead program is a PPAR alpha antagonist, and I'm sure almost everybody in this room is familiar with that pathway, but the other side of it, right? Agonists for phenylfibrates and everything. And that was interesting because when we would go try to talk to people about it, we would say, no. We're the other side of the field. We're an antagonist, and we believe, and it was definitely an academic commercial partnership there where we were working with an investigator, and he said, no one's looking at this in cancer, will you guys do it? So that's been an uphill slog to get people excited about that. Now we just came out with some early positive frontline data in liver, so heads are definitely turning on that, and it's kind of a we told you so. But then our second program was known target adjacent. So it's a dual EP24 antagonist, and everybody knows that the prostaglandin pathway is somehow implicated in cancer. I mean, any oncologist will tell you there's just that simmering data, but there's never been a really strong study. That one was easier to get with everybody because even though I think to our knowledge, we're the only dual 2 and 4 antagonist in clinical development. That just because it was adjacent to something that's really well known, it was easier to talk to people about it. But as soon as you went truly, truly novel, it was more of an uphill battle.
Yeah, but I think what Jim was describing in terms of target product profile and kind of the starting points of a med-chem campaign lead really naturally to kind of the de-risking translational work, because there's some trust. I mean, in the case of like rare fusion-driven cancers, oftentimes that's patient-derived models because we don't really have immortalized cancer cell lines. But nonetheless, you can go back and benchmark against existing drugs. But I think, Steve, what you're highlighting here with 2 novel targets, topic we definitely wanted to kind of get into a little bit is how should we be thinking about novel targets that are not activated oncogenes in terms of our ability to de-risk, kind of working from immortalized cancer cell lines, patient-derived lines, organoids, conventional xenografts, PDXs, right? Like kind of the conventional, if you will, cascade. For metabolism targets, if I'm allowed to call a PPAR gamma antagonist a metabolism target, certainly the agonists are, to a degree are. And then it's, kind of let's call it an inflammatory target, something in prostaglandin signaling. Just give us your sense of kind of, where we are in the field in terms of, using, what are, novel models, more complex models, and hopefully more patient-relevant models to kind of de-risk programs like that.
Yeah, I think it's whether you're an investor or a person inside a company that wants to be excited about your program, or you're trying to get, analysts and clinicians interested in what you're doing, on any of these novel targets, it is, sometimes we have to do really elegant work that is, edgy and saying, this is novel, so we need to do this. But it's funny, it always comes back to, do you work in vivo in the 3 or 4 models that we all want to see tumors, shrink in, right? And so when it's really, really novel, where it's not adjacent to some real estate where we're all comfortable, the preclinical package just has to be that much bigger. Right, so for example, back to the PPAR alpha, so what we've been told it's immunometabolism, we've been told there's a lot of really fun hyphenated words, but we have this beautiful, beautiful beta-catenin, liver cancer model where the molecule plus, it's called TPSD1120, crushed. I mean, it was a cure model, right? And so I remember I looked at those data when I was joining the company because we didn't have clinical data yet. And I said, wow, that's really impressive. I mean, I know there's a lot of healthy mice walking around, but this was just a really, really robust job. And now, fast forward to now, it was absolutely predictive of the liver cancer data we're seeing now. And we haven't seen the beta-catenin mutant subpopulation yet, which we're very excited to see, right? Because we all know how implicated that is. But I just think, just to sum it up, you just need that much of a bigger preclinical package than you do if you were going on a more trodden, like you said, more picked over target where people have seen a lot already.
I just want to underscore one word that you said before I hand it back to Jason. You used the word shrinkage. I mean, it does seem to me that in the market that we're in right now, particularly, tumor regression in an animal model, like it seems like a very consistent, not just ask, but demand.
And not prophylactic.
Yes, and
Yes.
somewhat established tumor.
Absolutely,
right, yes,
absolutely.
Yeah, like paying attention to the y-axis. How big was this tumor when you started the experiment? Right, I got it. Jason?
Yeah, no, it seems like when we looked at the pipeline for targeted oncology 3-4 years ago, maybe a third of programs were novel target. Now it's about half. So I think the answer, from what I'm hearing from you and just our assessment of the space, is that the answer is a little bit of both, right? There's opportunities in established targets, there's opportunities in novel targets. I guess as it pertains to sort of investing, behind programs that are going after novel targets, do you feel like there's better early stage tools to characterize these opportunities, or are investors really just being asked to underwrite higher risk, high upside opportunities?
You want me to start?
I'll comment on this. I previously worked at a company called Ignita, where we developed a next generation ROS and TRK inhibitor, so preceding Jim's work. And so there, it was a very investable hypothesis, kind of to some of the points that Keith made, that if this driver drug that protein, and it should work. And we as a precision oncology field have gotten somewhat addicted to that thesis due to its simplicity and easy provability. And we are showing that we can continue to make innovation, as Nuvalent is doing, or we're seeing with EGFR inhibitors like Tagrisso, or HER2 inhibitors now, the ADCs, even though these targets have been around for 20 years. However, I think it's incumbent on us as leaders in the precision oncology industry to look ahead and say, what's next? And I think we are going to have to wean ourselves off of the, if A is the driver, we're going to drug A, and go to what I consider kind of first derivative plays, where if A is the driver, we're going to drug B. So kind of like synthetic lethality, if BRCA, then PARP. And that's what our company, Boundless Bio, is trying to do. We're pursuing a class of oncodrivers, which are the oncogene amplifications, essentially too many copies of a gene. And these have been proven undruggable by the classic targeted therapies, by the kinase inhibitors. So we have had to find these first derivative targets and then build out the models and prove, so far preclinically, we've just entered the clinic, that this also can have an elegant simplicity, even though it is one derivative away from what we've previously done as a field. And one of the terms you used was robustness of preclinical data. For us, that means showing that the biological effect is consistent across multiple different settings. So for instance, we're developing a first-in-class CHEK1 inhibitor where we have shown that irrespective of tumor type and irrespective of oncogene amplification, the effect of inhibiting this target is the same across a multitude of different preclinical models. So I think that's one of the ways that we develop some belief, some religion from the investors. It still is not easy, though. They really are addicted right now to the if A, then A.
Maybe say a little bit more, Zach, about thinking about target selection. I mean, what you're going to roll up your sleeves and go after before getting to the heavy lift of first med chem and then what you described, which is like a large number of models to try to understand kind of how a new potential therapy works. I mean, are there computational methods that you feel like now give one an ability to kind of highlight a target as being a particular dependency, a potential dependency, because you don't know yet until you create tool like chemical matter to actually test that dependency, different than 5 and 10 years ago?
Yeah, I mean, we were able to perform hypothesis-free target identification by using things like the DEP map and Broad data, where we were able to characterize different tumor cell system models by amplification or not amplification. And we're particularly interested in something called extrachromosomal DNA, where the amplification does not occur in a genomic locus, but instead occurs on these distinct circular units of DNA that float in the nucleus. And so we were able to use some automation tools to characterize different cell lines and then look at every gene that was knocked down in the DEP map. We were able to perform our own genome-wide CRISPR knockdown studies, and even things like biochemical tool compound screens using known drug classes. But by using multiple different orthogonal methods, the preponderance of data across these different methodologies were all pointing to certain hotspot targets that seem to have this consistent biological effect across different oncogene amplification-driven models.
We'll get to this in a minute, but so there's some obvious congressional pricing challenges with small molecules. Can you talk a little bit about other therapeutic modalities? Small molecules has been the domain of precision oncology, but the ability to go after some of these novel approaches with other therapeutic modalities. I'll leave that open to the field.
We're a small molecule.
Yeah, all of us are small molecule guys.
Dyed in the wool, small molecule.
I'm a chemist by training. I don't know much beyond that. So.
Well, but to your point, though, when we chose our targets, we didn't pick them because they were small molecule targets. We picked them because we liked the biology and we thought that was going to translate. All of us on the team at Tempest had worked with vectors and antibodies and all other types of interventional modalities. Our 4 programs just ended up as small molecules. And I'm still a huge believer, notwithstanding what's happening in Washington.
I mean, I can say I'm excited about the degrader field. Like it's I don't know if you're characterizing that as small molecule still or not, but it's certainly an exciting field. I think it's going to be watching of interest with what happens when that field over the next few years. And obviously ADCs are very exciting these days as well, but our focus at Nuvalent has been traditionally on small molecule drug discovery and development.
Yeah, any drug that gets inside of cells, I think we're going to call small molecules for today's purposes. So maybe just give a little bit more of your sort of state of the union on degraders. First generation degraders, in preclinical models anyway, seem like we're in the realm of maybe 50 to 70% target downregulation ballpark. Can we point to target classes where that's enough, or is it the case that we need to kind of just hold our breath and wait and watch for more advances to be made, different degrader platforms, smaller molecules in some instances, to be able to get to 80, 90, 95% inhibition? Just kind of what's your take?
Yeah, it's certainly not my area of expertise, Keith, but I would maybe turn it back to the panel and say what is the appropriate level of degradation necessary? I personally feel that the field is in its infancy at this stage, and so there's a lot to learn. But
I'd defer to you on that. Yeah.
For us, it's been empirical that kinases might require a certain level of inhibition. Yeah. We're working on some other classes of drugs like kinesins and other things, and we haven't found any predictive value across the target spectrum as to what level of inhibition, or in some cases degradation, is required.
That's a good way to describe it. Actually, we have a TREX1 program in the STING pathway. We're trying to selectively activate STING, malignant tissue. And it's definitely that topic inside the company is very different than the 2 clinical programs. So it's empirical. I agree with you. Yeah.
So there was a lot of hype in the last few years over KRAS as a target, and I think the general consensus has been maybe more limited activity on a single agent basis, the need to sort of find more potent versions of KRAS that can be drugged more safely in combination with other ways to maximize the value of the KRAS pathway. Mindful that you guys don't have any ongoing KRAS-related programs, but just your thoughts on the target and the pathway forward, because that's obviously a high incidence target.
Yeah.
Go ahead, you can start.
Yeah, I'll start with just as a chemist, adagrasib and sotorasib are beautiful molecules, and it's kudos to those teams for the development of those compounds so efficiently and getting them to patients. It does look like the durability is somewhat lacking, particularly if you compare it to other targets that I know Nuvalent's focused on in the kinase space. That if you are drugging that particular target, you can drive deep, durable responses. I think the median duration of response in the first 2 approved therapies is in the 8 to 10 month range. And is that a molecule problem? Is it a mechanism problem? My personal feeling, it's a mechanism problem, not a molecule. I think those molecules are outstanding. So it is going to require a deeper understanding of the biology, combination approaches. That's my general take on where those stand.
And then there's a tension between how much work should we be putting in to make more potent molecules when most oncology therapies are combination, right? I mean, at some point, is it not worth, we're getting this marginal improvement, but you're going to end up in a doublet or a triplet or, more at some point anyway. So is that where we should go and put more of our resources?
And if I could add to that, right, I think there's a desire to solve all problems through better chemistry, but sometimes a more sophisticated understanding of the biology is what's necessitated. And one of the things we've seen with the MAP kinase pathway is single node intervention is insufficient. You often have to go down the vertical. BRAF and MEK is a good example where that's superior than to either BRAF or MEK alone. And then one of the things we're seeing with the KRAS pathway is it does lend itself, inhibition lends itself to bypass mechanisms, specifically through the amplification I was talking about earlier of other kind of adjacent onco drivers. And so to your point, I think we need to understand the biology better.
And it's not just the biology of the target, it's also the biology of what you're combining with.
Yeah.
Right? I mean, obviously cancer is phenomenally efficient at doing its job, and I go back to the combination therapy piece, right? When we were looking at what made the most sense to do with the PPAR alpha antagonist, going with a VEGF or a TKI, and then either a PD-PD-L1, preferably a PD-1, but we did it with a PD-L1, made so much sense. So we had target logic, we had potential subpopulation driver mutation logic, and then we had MOA of other drugs target, right? And so we've had that conversation. Should we have gone further with our PPAR alpha antagonist and made it more potent? And then we decided, maybe as franchise management and backup, but the real thing was just to find the right therapy in combination. And we're in a triplet now.
But this gets back to the issue of the challenges with novel targets in terms of understanding how much one can rely on preclinical models. When you have human beings with a fusion-driven cancers generally being some of the most genetically simple tumors there are, where you see on-target resistance mutations as the dominant theme, and very specific ones that very logically, don't allow existing drugs to bind. Right, I think then obviously, roll up your sleeves, more chemistry work. To trust that, within pathway bypass or compensatory mechanisms and other pathways in model systems are what humans, what's going to happen in humans. I think this comes to the point that I was hoping we'd leave a little time for, which is sort of moving to higher order drug development, as in combination development. Particularly thinking about novel combinations is the highest degree of difficulty for sure. Once you've got a drug on the market and it's got some effect, like the KRAS G12C inhibitors being a good example, then the idea of building on those is a little bit more terra firma. But when you've got a drug in your hands that does produce an effect in preclinical models, you think you can understand in model systems what kind of the, let's call it bypass, mechanism is, where you need a different, another drug. I guess I'm interested particularly in this environment that we're in now, but I said this, 2019, 2020 as well. We're having a hard time as a field in oncology moving to the development of 2-drug regimens, and certainly 3-drug, 4-drug regimens.
You mean novel, or?
Yeah,
okay.
it's novel. Well, and by novel, I don't entirely mean that like both drugs are at a synchronous moment in drug development necessarily, but certainly not a marketed product, right? So like the foot in the door, let's say primary vulnerability targeted agent, just doesn't carry enough weight, right? But that you've got in your hands data that suggests, well, we understand that we can drug, a second mechanism as well. This is where I think biology points us. This is like simply put, like primary vulnerabilities that are going to cause tumor regressions in models and deep and durable response in human beings. We want as many of those as we can come up with, and we're obviously, to Zach's point, moving beyond oncogene targets to a broader set of, just call them non-mutated targets, broadly speaking. But if certain cancers can't be wrestled down without a 2-drug approach to even produce a 1-drug-like effect, this is, to me, this is the kind of the continued moment of paralysis, and maybe we just need fundamentally different financial markets before one can even try to take risk in this territory. But
Yeah.
I'll comment on that because we're grappling with this very issue in our development plan. So we are developing some of our novel drugs in combination with other drugs. And for our very first clinical study, we're going to take our novel compound and pair it with approved drugs. And the way we're going about doing that development is supply agreements with pharma, where they simply provide us the drug and we bear all the risk. However, we are also working on some novel combos where the other drug is not yet approved. And in that case, we're actually entering into, or we have entered into, preclinical collaborations with pharma to establish that evidence base where now both sides are bearing more risks. So that's how we think about risk diversifying, is how much we're asking our pharma partners to bear.
And just quickly, is this, I mean, maybe it's too early to tell, but because you're poking around in preclinical models, but is this a randomized phase 2 proof of concept like default?
Right.
Which is, certainly in the venture-backed biotech world is like, that's a phrase that gives people hives.
Right.
It's rare, yeah.
Or not.
So the answer is no, and perhaps not always satisfying to investors, but to me personally satisfying and based on evidence, which is what we are doing is we are combining an approved drug in a setting where it has not been approved because it's already outright failed with our novel drug. So the evidence base for us is if you take an EGFR inhibitor into an EGFR amplified setting, it has maybe a 10 to 15% response rate and has not warranted approval. So if you now combine it with our novel agent and show a 40% response rate, it would be very hard to argue that that was due to the EGFR inhibitor alone. And so that's how we initially build the evidence for proof of concept that would then lead to a discussion with FDA about what is required in a potential randomized study, which brings up interesting questions of, is the control arm a single agent drug that has already been proven not to work? Like is that an ethical thing to do?
Right. I think also just intrinsic in what you said, by the way, though, is what I sometimes phrase as at least aiming for hitting doubles and not bunt singles in terms of like what you're looking for to constitute proof of concept when thinking about combinations.
Absolutely.
Yeah, big effects, if not home runs.
Last week was at ASCO and saw Dr. Kantarjian's presentation on the evolution of leukemia lymphoma treatment. And the work, I think a good example of what you're getting at is the work by BTK and BCL2 combinations, novel combinations, which are driving patients to MRD negativity and frontline CLL. Just amazing progress in the last decade of direct novel combination drug development. Really changing the treatment paradigm for cancer patients. So I thought it was maybe apropos of what you're trying to get at.
Yeah, absolutely.
It just becomes a financial problem, right? So to your point on how are we going to address the biology and the way the tumors work, absolutely we could sit down and go novel, right? I mean, that would be wonderful, but especially, I know we don't want to go down capital markets rabbit holes, but in this market, it would be really tough, right? For me to sit with the team and say, let's do that, even though that is the best scientific approach. We can't finance that right now because even though there is a path with FDA, I mean, the sum of the parts conversation is just, becomes prohibitive.
Can you talk a little bit about the overlay with Project Optimus, right? Because now the ask is, fully interrogate. I think
Yeah.
FDA at ASCO 2 years ago was like, we don't want you just pushing max tolerated dose just to drive an ORR in a single agent. We now want to see kind of interrogate doses, possibly see RCTs. And now you're trying to prove contribution of parts with doublets. So it seems like a very complicated puzzle to solve for.
Yeah, we've been scared of Optimus for a while because it's actually been around. They've had a single-page website for years. It just never came up. And then I was talking randomly to one of my neighbors on a street, and who works at Large Pharma, and they said they had just dropped a heme program because FDA was asking them for too much multiple dosing and schedule, and the NPV didn't work anymore, and they decided to drop the program. And this is pre-IRA. Right, and so we ended up doing some of it at Tempest just it was fear. We didn't think it was necessarily necessary. It was really just to address the regulatory piece. So yeah, I don't know how you guys have looked at it.
Maybe I'll take the unpopular opinion, but I think it's well needed. There are so many drugs that are in late development or even approved where it's clear the drug is not well tolerated at the dose that's being developed at. You see nearly half the patients with dose interruptions due to adverse events, and then patients could progress during their dose interruptions. There's a specific approved drug in the ALK space that we hear from physicians that they don't prescribe the approved dose. They actually prescribe the dose below or even 2 dose levels below because the patients can tolerate it better. So I think the impetus to focus on getting the drug right early development is an important one, and I understand the initiative. We all also are very interested in speed and getting drugs to patients as fast as possible. So trying to balance how quickly you can learn about the pharmacokinetics, the pharmacodynamics, the safety, the activity in phase 1, so you can get that to pivotal studies, obviously is what we're trying to do. But it's just as important to think about making sure you're getting the dose right in that early development.
Yeah, I'll build on that a little bit. I agree that scientifically, I think it's well justified, so don't necessarily have fear about the basis of it. I think what's been a challenge to the industry is right now it's not particularly prescriptive, and there's not a set guidance on this is the exact number of patients you need to have at each dose cohort, or here's the log-fold change in dose cohorts required. So I think that uncertainty probably poses fear to some. However, in uncertainty is also opportunity, meaning that sometimes as a small biotech, you can take advantage of that and ask for something a little bit more bespoke or that works with a biotech's, financing ability. So we're not so concerned about it. We generally find FDA to be rather reasonable if your justification is well reasoned.
Well, I think combining maybe 2 recent points you both made. This is a crosstalk between efficacy and toxicity. So in swinging for doubles and greater. In other words, drugs that have an evidence base to suggest a pretty big effect in a high unmet need area, where the dose reduction rate is 20%, not 40, 50%, which, as you rightly point out, if you trace out drug approvals and the number of drugs that have that kind of high dose reduction rate, that's a pretty recent thing. You dial that back, short of randomization, but just you can demonstrate in a large enough safety database that's the case. With a big therapeutic effect, my strong sense is that data set does not struggle on the path to at least accelerated approval and making those therapies available to patients. But to be determined as to which of the therapies that which profile is subject to the greatest scrutiny, the most investigation needed to this point, I think.
Yeah, maybe just along those lines, do you feel like the accelerated approval pathway is sort of gone by the wayside, right? As a lot of categories have advanced, and now with the overlay of IRA, perhaps indication sequencing becomes a greater aspect of the thought process. So you could go after accelerated and maybe be a very late line niche opportunity. Then you start the clock running on your molecule. So maybe how are you guys thinking about those challenges, mindful that maybe there's a lot of uncertainties in front of us in the next few years as this gets sorted out?
Yeah, we're actually right there with the liver, the PPAR alpha program. So as I mentioned, we're looking forward to getting beta-catenin subpopulation data. That could be a breakthrough and then an accelerated approval path. And so we're already structuring the randomized phase 3, and we were talking about like if the signal is strong enough, would we put in a front piece that's an accelerated approval piece and then use up some alpha and then go into the larger population, because all of our data right now are in the larger population. So I don't think it's dead as an option. I'm on the board of Bio, and when this was all happening at FDA, we had all these conversations around what is reasonable for FDA to expect of us. And it was funny, it was all the CEOs of the biotech companies who were saying to the lobbyists, no, we actually owe them a confirmatory study. Like, we were the ones saying it's reasonable for FDA to actually expect us to keep our promise. What wasn't on the table is really where we are now, which is single arm accelerated approval, which back 10 years ago, I worked on a proteasome inhibitor in multiple myeloma, and that got approved on a straight, old-fashioned single-arm study, and it's a drug, so there wasn't a risk of it not happening, and it's helping a lot of patients now. But it's a foregone conclusion that we're going to be doing a randomized lead into an overall population.
My limited experience is that it's somewhat contingent on disease prevalence and therefore feasibility of running a timely randomized study. So in the NTRK space, which Keith and I have experience with, as well as the ROS1 space, you can successfully argue for an accelerated approval based on a single-arm study, because it can be very difficult to run a randomized study. Both of the recent KRAS approvals were accelerated approvals based on single-arm studies, but where they had begun enrolling the randomized study, because of course KRAS is a more prevalent biomarker and driver. So I suspect that logic will continue to hold up, that if you have a very prevalent driver and you try to say to the FDA, we just want a single-arm approval because it's not feasible to run a randomized study, they might look at us kind of cross-eyed.
Yeah, I agree with Zach. I also would say that the FDA's interest in randomized studies is not new. This has always been the push for the last 20 plus years I've been in the industry. Any single arm trial you bring to them, they will tell you they prefer a randomized study. It's where a randomized study might not be appropriate, because it's not obvious what their comparator should be or what the design should be, or, as you mentioned, Zach, a limited patient population. So if you have a well-designed single-arm study in a clear area of medical need, and you have really transformative data, I think there's an opportunity to engage with regulators about an accelerated path of a single-arm study. But those things have to line up, and as you mentioned, Steve, having the confirmatory study underway, I think is a point of emphasis from regulators these days, and certainly something that all of us should be paying attention to.
Yeah, it was surprising how many companies just didn't run confirmatory studies. I was shocked to see the data. No one here.
Yeah, exactly. Well, maybe just one quick nuance to add. I mean, ROS is relatively, almost completely restricted to non-small cell lung cancer, though within histology. Increasingly, we see evidence, not just with genetically altered targets, but others, that their dependency in cancer distributes across cancer types quite unevenly oftentimes, but some of them are sort of sparsely distributed in a way where doing a randomized study across in a tumor agnostic way, God help us, or in 6 different tumor types, right, becomes like challenging to imagine from a feasibility, statistical rigor perspective, right? That this is, I think, goes to Zach's point about kind of the commonness as one axis, but then also just kind of the context in which you find an alteration dependency or whatever molecular feature that becomes your defined population.
Yeah, so in our ongoing ROS1 and ALK studies, we do allow other tumor types outside of non-small cell lung cancer in the phase 1, and also we have phase 2 exploratory cohorts in those other tumor types. The challenges there are, one, are patients being sequenced in those other tumor types, right? So you can find the patients. And then 2, what is the regulatory path? It's, is a tumor agnostic development strategy appropriate? I would say most likely not in those particular tumor types. I think that in the ALK space, you've seen crizotinib do a study in lymphoma patients, got an approval, a specifically small pediatric lymphoma patient population. There are obviously the examples in the TRK space. I know that some of our panelists are quite familiar with, and those are beautiful examples of tumor agnostic drug development. But I don't know if it's a one size fits all across disease drivers.
I'll just add a quick bit to that. So if you are doing tumor agnostic with the TRAC or others, then you have, obviously the error bars get wider because the expected response of chemo and colorectal versus lung versus salivary gland tumor are going to be different. So if you have wider variable error bars on what you're trying to beat, it just means your treatment effect has to be that much larger to make it obvious that it's a clear win.
Well said. Close this out.
Well, we've got about 10 seconds left, so maybe I'll squeeze in a question from the field here. So studies have shown that diet plays a significant role in the development of cancers. In your opinion, to what degree does nutrition play a role in oncology and the extent to which outcomes can be improved by modifying diet? I don't know if this has much relevance on the precision onc side, but.
Yeah, I'm just thinking about our targets. It's not. No, it's a chemistry question. Can your molecules hit their target without food? But that's not what the question means.
The role of diet. I mean, early in drug development, all of us will run food effect studies to understand the impact of food on the ADME properties of our drugs, and that guides the clinical design protocols, for later stage development. So that's typically how it's done. Usually you have an understanding from preclinical models, preclinical experiments as well, but the early development, you're going to run that food effect study to understand that about your molecule before putting it into broader studies.
Just maybe I'll close this out with this comment. The observations that relate diet to therapeutic effects in cancer, pretty large-scale studies, which is typically kind of a post-marketing discovery as opposed to the pool in which you all and your companies are innovating in terms of early smaller scale development, where it's harder to make those observations, I suppose. But with that said, we have exhausted our time with a great discussion. We really appreciate all the contributions from the panelists. Thank you all for your attention, and we move on.
Thank you.
Thank you.